Hello, I'm Jan Zrimec and I'm a postdoc in the group of Alexei Zelezniak at Chalmers University of Technology. And I'll present our recent research regarding learning the regulatory grammar of DNA for protein expression engineering. So, understanding the regulatory code of gene expression and being able to manipulate it can enable us to fine-tune the functioning of cells, and this is important for solving problems in medicine as well as biotechnology. In the process of expression of DNA to RNA to protein, it's been shown in multiple organisms that actually protein abundance is in strong agreement with mRNA levels. This suggests that actually transcription is the major determinant of protein abundance. So here we focused on transcription and we obtained a large amount, so almost all available RNA sequencing experiment covering the majority of conditions. And when we sort the protein coding genes according to their average gene expression level, we find that the variation of expression per gene across the conditions, this variation is actually much smaller than the whole dynamic range of expression levels. So this means that genes that are highly expressed across most conditions will likely be highly expressed, and vice versa for lowly expressed genes. But it also shows that there's this basal average gene expression level is conserved for each gene and is likely encoded in the DNA sequence. So for this, we needed modeling to test this. We needed a modeling framework that is able to extract information from DNA sequence. And luckily, in past years, there's been a lot of development on these deep neural networks. And they're able, for instance, to decode information in very complex images and translate it to human understandable language. And these same neural networks, for instance, convolutional neural networks, can also be used for sequence analysis. They can learn to recognize the occurrence of different motifs or groups of motifs. And recurrent neural networks can actually learn the sequential information of these motifs in a DNA sequence. But recurrent neural networks are actually very hard to train, and certain tweaks with convolutional neural networks can achieve similar results. So here we just used stacks of convolutional neural networks with additional dense, so classical network layers. So for training the models, as explanatory input variables, we used sequence upstream and downstream of the coding gene sequence. This included promoters, untranslated regions, as well as terminators, and spanned altogether over 2,000 base pairs of DNA sequence. We also used the codon usage of codon frequencies of coding regions, as well as additional variables related to mRNA stability. So the trained models with this data from yeast were able to explain over 82% of the variation of gene expression levels. And we also then used other model organisms spanning from bacteria to human, so a very wide range of genetic complexity. And we found that on average, 60% of the gene expression level variation could be explained from DNA sequence alone. So we further analyzed and compared the deep models with shallow ones, so classical machine learning models, as well as analyzed how different regions contribute to prediction of expression levels. So we found two important points: that compared to shallow models, the deep learning approach enables us to extract much more information from the regulatory regions, and also that all of these regions jointly contribute to prediction of expression levels. So each region carries a certain amount of course, overlapping information plus new orthogonal information that's important for prediction. So to analyze this further, we use orthologous genes from 14 related fungi species and analyze the mutation rates in promoter, terminator, and coding regions. And we found strong evidence that promoters and terminators actually co-evolve with the coding regions. So this shows altogether that the gene that the gene regulatory structure that we are analyzing here is not only interacting, but is a co-evolving unit. So next we wanted to extract and analyze the DNA regulatory grammar that these models learned. And for this, we used a simple approach, sliding window occlusions of input data, and then we measured the perturbations of the output. So we determined the relevance of each occlusion for predicting the output. So with this, we were able to determine the most important regions of DNA and from them reconstruct the DNA motifs. And in this set of motifs, we found that we recovered a lot of the known and expected regulatory grammar of yeast, spanning all the different regions. So this included transcription factor binding site, cosec sequences, as well as other known motifs, for instance from terminator regions. And after that, we also performed association rule analysis, which is actually market basket analysis. So we treated each gene as a basket and determined the co-occurrence of motifs across the different genes. And what this showed is that over 8-fold more co-occurring motifs were found across the different regulatory regions than in any single region alone. So an example of this is shown here for like four genes that have a common transcription factor binding site motif in their promoters, so marked blue, and they also carry additional motifs in the other regions, and these motifs actually repurpose the transcription factor motif and define completely different levels of expression level, covering basically the whole range of expression levels. So then we analyzed how much of this whole range of gene expression levels we can recover either with single motifs or with motif co-occurrence rules. And whereas with motifs we can recover 57%, with the co-occurrence rules we're actually able to recover 84%. So that's going towards defining the whole range of gene expression levels. So finally, we asked whether we can use these models of DNA regulatory grammar to guide gene expression engineering. So we considered first that the way people usually approach fine-tuning gene expression is to focus just on a certain region, like the promoter, and swap it out for certain known strong or weak promoters. And they don't really take into account any of the other regions. So here we could exploit our models and analyze, for instance, if we keep the promoters fixed, so just native promoters of genes, and vary the terminators with all the different terminators that we have in our data set, we find that actually each gene, so in each promoter gene pair, has a large amount of regulatory freedom, so covering over 2 orders of magnitude for gene expression levels, just by swapping out for different terminators. And we achieved very similar results also when keeping the terminators fixed and then varying across different promoters. So we then experimentally verified this. We created constructs with native promoters and predicted strong and weak terminators. And we measured that actually on average, indeed these combinations could increase or decrease the expression levels by 20%. And we also found certain much stronger combinations. So what this shows is that actually we can guide gene expression engineering just by learning from the native genome and without requiring any synthetic approaches. So an early version of this work is available on bioRxiv, and hopefully the final version will soon be published. And it was a collaboration with different groups like Computer Science, Big Data, University of Gothenburg, and of course SysBio. And most of these people are also co-authors of the above study. I especially want to thank Alexei Zelezniak for giving me the opportunity to work on this very interesting research. And thank you for your attention.